Thank you, Geraldine, and thanks a lot to Sakera for putting this event together. I think it's great, and it's nice to see familiar faces every year. My name is Alvaro Martinez Barrio. I'm the Chief Data Analysis Officer at Pixelgen Technologies, and today I'm going to talk about 3D representation of single cells without a microscope. Yes, this works. Okay, so in the past summit, we have been both in Barcelona and in Boston introducing first NF-Core Pixelator. This is our pipeline that in Nextflow and the NF-Core project, we have put together the first company, to my knowledge, that did it from the very beginning as a public project in order to process or data type, or very specific data type. Then also we have highlighted the applications that you can use for MPX, and currently there are many labs using them. Then also, in that case, Nextflow and NF-Core has enabled us to deliver our data analysis capabilities to a faster pace to our customers. Our MPX methods paper was published as of May this year. This is published in Nature Methods, and it's open source, so you can actually browse it. The nitty-gritty details of the methods are covered there, so I'm not going to touch upon that. But currently, as I said, there are many customers that are using our technology in different labs, and most often even in parallel projects. And the applications are very diverse, but this is not what we are going to be talking about today. Just to reiterate from previous talks, what MPX or molecular pixelation is able to achieve is probably the unique high throughput method that is simultaneously counting proteins and the relative spatial positioning of these proteins in the cell surface. What this enables also is that you can query proteins that are in pairs or in groups together and find co-localization patterns of those proteins. Recently, as of last July, we have also improved for chemistry what we call MPX version 2. And you can browse that tech note that is referred there in that QR barcode, and it's actually covering the aspects that are most improved, namely the number of proteins that we detect. Also, we have improved the signal-to-noise ratio, and also we have reduced drastically the isotope control level background. Okay. So to the topic today, what are the key principles that allow us to display these proteins in 3D? So basically there are two. There are these DNA-encoded barcodes that are able to position proteins at subcellular resolution and then putting them together into thousands of cell graphs. And the readout is also special because we don't use any microscopy. Our readout is based on next-generation sequencing technology. So how do we do this, you might wonder? We have this new stage since version 1.20 of our pipeline called layout, and what is able to do that stage, it contains the logic of converting MPX cell graphs into 3D spatial graphs. So the first challenge that we faced, it was how to convert spatial graphs that contain no 3D position information available. And then we had to find a method that could be highly performant to generate these coordinates for 1,000 cells that are usually the throughput of our MPX experiments. Okay, so what assumptions and what are kind of like the information that we had at hand? At the core, an MPX experiment is represented by an edge list, and this edge list contains the following information. In each row, basically, there is a protein barcode with a unique molecular identifier and an edge between two DNA-encoded pixels, the A pixel and the B pixel. And that means that those two pixels are in close proximity in the cell surface. And when you form those particular graphs, those are named in the literature as spatial graphs. And the field of graph visualization is vast. There are a lot of techniques there, but because graphs haven't been very large, they don't really cover well spatial graphs. They scale often poorly to large graphs. So we had to look around, and we found this algorithm called multidimensional scaling, or MDS for short. And what MDS does is that it takes a distance matrix of the different set of endpoints and then transform them into endpoints on a Cartesian coordinate space. Okay, so let's do a toy example so you can refer home. And I'm going to go through this very quickly and skip a lot of steps and assumptions that are not reflected in this short presentation. So we take the set of 2D points, distribute it, and then we connect them to the closest nodes. This is a very well-connected graph, so thereby we form a spatial graph. And then when you are drawing these pairwise distances, you find the shortest path. And in this basic toy example, what you count is the number of hops that you are doing in that spatial graph, and just computing that as the pairwise distance between those two nodes. What is an interesting realization here in the spatial graphs, both in 2D and 3D, is that the Euclidean distance of the graph, as measured in this Cartesian space, correlates really well with actually the shortest path distances. So we take this distance matrix that we have calculated now between all the points. We use MDS, and then we reconstruct in this toy example a quite good representation of it was the original point distribution of the mock-up example. So there is a major drawback of this approach, and the drawback is that there are two steps that are very computationally expensive and consume huge amounts of memory for large graphs. So we had to find a solution for that. And what we found is that by drawing what is called pivot points, by taking a subset of random points in the graph and just using those pivot points in order to calculate the distances to all the other points in the graph, then you could achieve this in a feasible amount of time and memory for the 1,000 cells that normally run in our experiments. So PMDS performs pretty well in our toy 2D spatial graph and how it looks like in a real data point of an MPX graph. So this is how it looks like. So it looks pretty well, but I think this is not maybe like a spheric-like cell representation. You might see some noise there. So the issue is how to tackle these noise edges. So how we adjust the spatial noise in MPX graphs, and the trick to that is to use edge weights. And I'm skipping a lot of material here in this particular 3 slides. So we update the edge weights, and the edge weights is basically the number of unique protein molecules that are contained within each pair of pixel A's and pixel B's that reflect a lot of spatial information, how close to each other they are. And then with the edge weights strategy, with the updated penalizations that we create for badly connected nodes and also edges linking too far apart nodes, we are able to get the right representation of the same picture. These also help us to represent better the especially interesting regions that we are able to detect, like for instance, this CD82 polarity event in a PHA-stimulated T cell. So it's more visual appealing, and then you understand that there is a ton of CD82 protein in that particular area of that cell. Right, so there is a lot of video information. You can follow us in our YouTube channel. We are creating a lot of new material, and we hope to actually record a longer talk of the material that I'm presenting today with all levels of details of the different steps that I'm jumping today. And we love open source as well at Pixelgen. So basically, we have a lot of expansion in tutorials available to how better analyze single cells. And the last ones there, like the 3D cell visualization and the advanced topics, spatial patterns tutorial will allow you to do the same kind of visualizations that I'm actually presenting here today. We have also 5 public data sets that you can use in your research. They are licensed out in very friendly terms. And then the last one actually contains the demonstration of our version 2 chemistry. So people say that it takes a village, and it's true. So I want to acknowledge all the different pixelators that have created and put this technology on the hands of our customers. They are great, and it's really fun to work with them. We are several of us here in this summit, so if you feel curious about MPX and Pixelgen, just come and chat with us. We are super friendly. Yeah, and this is maybe my last slide. I will skip.